Yes, sir.
Okay, I have started recording our lecture. So today is our third lecture. Here we're going to see about the various modification and the structures that are present in the plasma membrane. The last class we focused on the fluid mosaic model of plasma
Fluid
membrane,
music model.
and we discussed about what will happen, that is what is the main reason for the membrane fluidity and what are the factors by which the membrane fluidity is highly affected. So we have seen all this in the last class. About the major functions will be
Yes,
seen
sir.
one by one. Now we can go to see the differentiation of the plasma membrane. So there are a lot of modification we can see in the plasma membrane. First, when I take about a
Sir,
procap,
you not teach the sir.
yes ma,
Sir, you not teach the lipid raft, sir.
lipid raft is the lipid arrangement that will be focusing with respect to the functions. Pooja,
Sir, 1 minute, sir.
what happened? You're not
1 minute,
able to
sir.
hear me?
Can you hear? Okay. Now can you hear me, sir?
Yeah, any network lag better there?
No, no, sir. I was not able to hear you properly. That's all.
Is it so? Let me check my net here.
No, no, no. It was my. The problem was within myself, sir. Now it is okay.
Now it's okay, right? If you have any network issue, you can tell me. Lipid rafts about the lipid arrangements only, but about raft means
Okay.
how they actually are arranged to perform some functions. That will be focusing on the functions
Okay.
of plasma membrane respect to the cell recognition and information are there now. For that, we require GPR linked anchored protein. We have some
Okay.
linkers which joins your protein into the lipid layer, how they are joined. All this will be focusing with respect to the functions. Okay ma.
Okay, so you're saying that the lipid rafts is attachment of lipids to the attachment of proteins of
That is
proteins
attachment of
carbohydrates to the
biomolecules into the lipids. Okay, oh, do one thing, I'll take you lipid raft, then I'll go for the cilia or flagella. Oh, that will be fine, I think so. Okay, better.
yes, sir.
Will that be fine?
Yes, sir.
Okay. I thought that to take lipid rafts as one of the functions with respect to the plasma membrane, no problem. So, what are this lipid
Okay.
rafts means? These are some active centers. These are some active center of the membrane. Already, the lipid is actually a highly reactive center, but in that we have some lipid center. The lipid center includes, they are made up of cholesterol, they're made up of some proteins, which are your peripheral proteins, and we have some conjugated lipids. I told you what is conjugated lipid. Do you remember what is conjugated lipid? No idea.
Um.
Okay, glycosphingolipid. To a simple lipid, if I add another complex macromolecule or other biomolecule when I add, it will become a conjugated or complex lipid, right? Right?
Yes, sir.
So
Yes,
these are
sir.
some of the active centers in the membrane. That is the specific areas as given here. It is enriched with cholesterol. It is enriched with sphingolipids. But how they are joined in by some linker molecules. So where they are located? They are located in the plasma membrane, and they have some associated area also, which is the Golgi body. What could be the common functions? Number one, cell signaling. The two cells have to communicate with each other, right? The topic is given here. I'm not introducing you there.
Yes,
Here
sir.
we have junctions, that is intercellular junctions adhering desmosis gap. I thought to introduce that little bit later, but anyway we'll introduce now itself. There is some cell signaling. So what happens here? Generally, if you look about the outer layer of a plasma membrane, in that what happens? These maybe GPI anchored protein or some of the cholesterol or some of the, mostly it is actually GPI anchored protein. This GPI anchored protein which acts like a receptors. This GPA anchored protein, they act like a receptor. What is the role of receptor? For example, if a cell is having the following structure, say I have a cell here, say it's cell A, and this is my cell B. So in the plasma membrane, we are as a modified glycoproteins are there or glycolipids are there, but we are taking a GPA anchored protein. This GPA anchored protein is going to act like a receptor. Imagine that the receptor is
Mm,
having function like this. One is having like a V shape, others having like a half semicircle shape. If I have some molecules,
mm-hmm.
this molecule will enter into cell A or it will enter into cell B where they can go and bind.
They enter into cell B, sir, because of
Because
the GP anchored protein.
yeah, we have some receptor in this. Receptors are involved in recognizing the foreign particles. Purinjithingalama?
Mm,
That is the
mm.
role of this GPI anchored protein. Next is actually
Okay, sir.
will be having this cholesterol and transmembrane glycoproteins. Lot of proteins are there. I'll tell you in a simple way.
Okay.
I have an
Mm,
organism here. Say that it is your WBC. If it is your WBC, the WBC when they engulf any foreign particles, if this is any
mm,
antigen, if they are encountering into your body, the next phase, your false feet will be formed by our WBC.
mm,
In this false feet, the foreign particle will be embedded. Following this, the foreign particle will be completely covered by this membrane. This process is called as endocytosis. Cell will engulf the foreign material. To engulf and to form the structure, we don't require the proteins, but along with some proteins, we require a glycolipid because to change their shape, we require glycolipid. Along with the glycolipid, we require a protein which is a oligosaccharide. That is how the glycolipids or anything is joined. Oligosaccharide, that means sugars are there. So they are joined by sugar molecules. The sugar molecule acts like a receptor. Not only that, are you able to understand?
sir.
Oligosaccharide means sugar. So sugar units are
Yes, sir.
combining this GPI anchored protein. Along with glycolipid, they help in recognizing the foreign particle and consuming the foreign particle by a process called as endocytosis. Endo means inside, cytosis means cellular movement. A cell is eating in another cell.
Okay. Sir,
Ah.
so you're saying that a GP anchored protein polysaccharide linker are all acting like a receptor.
No, only GPA anchored protein will act like a receptor. But how this
Okay.
proteins can be bound into your lipid layer by an oligosaccharide linker.
Okay, okay.
And this
The
glycolipid is
connection
yes,
between
yes, 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 please.
the connection between a GP anchored protein and with the glycerol head is possible through a polysaccharide
Yeah, it is
linker.
joined by an oligosaccharide linker. Just it's a connecting link, like a glue. It will just go and connect. But lipids straight
Okay,
away they
sir.
are connected here. But you know this outer membrane is hydrophilic. Here one glycolipid is
Yes,
there.
sir.
So one glucose molecule and
Mm-hmm.
lipid is also joined. So glyco stands for again one sugar. So this same
Yes, sir.
oligosaccharide linkers joins in another lipid because glyco when the
Okay.
term comes, glyco stands for glucose, which is actually a sugar molecule. So here we have the lipid. So the sugar molecule is joined to the membrane again by a sugar molecule linker. Got it?
Yes, yes, sir.
Similarly, we have transmembrane proteins. These transmembrane protein also will act like an, for example, this is actually called as antimicrobial response. Antimicrobial response by Endocytosis. When an antigen or a pathogen encounters your body, a cell have to engulf it. So this also is mostly associated with your membrane. Are you able to understand what is this raft?
Yes,
Raft
sir.
is, yeah, it's actually
Yes,
a recognizing molecules, and they mostly help in cell signaling and cell recognition. So they help in cell signaling, plus they help in cell recognition.
sir, I have a doubt, sir.
Hmm.
Like that, you explained that attachment of biomolecule to the glycerol part with the help of a sugar. That area is called as liquid draft, sir.
Yeah, entire thing. That is, and all our functions are coordinated, right?
Mm-hmm.
Okay. What is lipid raft? It is a active center of a membrane, which is made up
Membrane.
of cholesterol, proteins, and the complex lipids. Okay,
It, yes, sir.
and lipid rafts are nothing but how they are joined into the membrane means the lipid rafts are nothing but these are the active center. The active centers are joined by a sugar molecule into the membrane. The main function is cell signaling, cell recognition, and antimicrobial response. Purunchama?
Okay, sir. So, yes, sir. I'll repeat it, sir. So, lipid rafts is it is the center. It is the main center where attachment of cholesterol, peripheral protein, conjugated lipid with another lipid layer through and through a glucose molecule is called as lipid
Yeah.
rafts.
So your definition comes like
Okay.
this. It is a attachment of biomolecules in the plasma membrane. Am I correct?
Yes, sir. In the plasma membrane,
Hmm.
it takes place at certain places which is rich in cholesterol, peripheral proteins, and conjugated lipids, sir.
That is, yeah, that is the active centers of the membrane are made up of the following things. To this, some of the biomolecules are joined to bring out the following actions. Okay.
Yes, sir. Yes, sir.
Now, so main functions is of course we're going to see one functions, that is compartmentalization, barrier, exchange of information, cellular movements. But don't worry, we're going to see all the functions one by one. Okay.
Yes,
First,
sir.
is cell movement is also there. So let us focus one by one. So first topic we're going to focus is one of the modification is cilia. Okay. Can you hear me?
Yes, sir.
Yeah. Now I'm taking a microorganism. Okay, I'll take a prokaryotic cell. I want the cell to move from one place to another, but there's no locomotory organ. That means they are called as non-motile. If they want to attain motility, I need to provide some structures, which is a modification of plasma membrane, that is a hair-like structures. If they have the following structures, they are called as motile bacterium. If I take another organism, this is called paramecium. You know what is paramecium, right?
Yes, sir.
It's a protozoa. This paramecium also having
Uh,
one structures outside their body, which gives them the locomotion. At the same time, I have another organism. It's called as a connecting link between plant and animal kingdom. What is that organism called as? A connecting link
so
between
can you repeat it again, sir?
an organism which is known as connecting link between plant and animal kingdom. What that is called as? Option A, Euglena. Option B, slime molds. It's called as Euglena.
Euglena?
Yes, it's called Euglena. In the Euglena also for locomotion, they have cilia or flagella. So these are some of the modified structure which is there in the membrane of the plasma membrane, that is called as cilia.
Hmm.
Got it?
Yes, so cilia, because these things you have mentioned, they are called as prokaryotes. Actually, they are not motile. In order to have a locomotive movement, a modification of plasma membrane takes place that is called as cilia.
Yes.
Okay.
Got it?
Yes, sir.
Okay, so we have got a cilia now. So I told you where are the various location of cilia. This is also a cilia, or they may have a flagella too. In case of here, they have
Okay.
a flagella. Flagella also is a modification only, but let us focus only on cilia alone. So what is the cilia? It is actually a cytoplasmic extension. So they where
Mm.
they arise from the base of a, come on,
Plasma membrane.
yes.
Cytoplasm.
They are extension beta. So where they are originated from means they are actually originated from an important body. Okay, in another way you can see it is a small cytoplasmic extension, because in this the cytoplasmic extension means if I draw this as a membrane part, let me take one eraser. So the contents of the cytoplasm also protrudes along with the cilia, right? So it is an extension of your plasma membrane, which is purely having the cytoplasmic extension also. Averagely, they come into 7 to 10 micrometer. What is 1,000 micrometer is equal to?
Sir, I have no idea, sir.
So you don't know the measurements, beta? Micro into millimeter.
I know the measurements, but I have read it, sir, but now I don't remember it, sir.
Okay, I'll tell you. Um,
Okay.
I'm taking some measurement. Okay.
Okay.
Okay. Will we go with okay. This is 1 micrometer. The 1 micrometer comparable can be compared with 1,000 millimeter. So it is 1/1,000 of a millimeter is 1 micrometer. If you take about a scale beta, 10 millimeter makes 1 centimeter. So this individual unit is,
Sir.
so 1, 2, 3, 4, 5, 6, 7, 8, 9. So if I take an individual unit, this is 1, which is 1 millimeter. So micrometer is 1/1000s of a millimeter. It's
Thousand.
very tiny structure, right? Very, 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 very fine, tiny. Okay.
Yes, sir.
So புரிஞ்ச எவ்வளோ டிஃபரன்ஸ்னு இந்த டாட் எடுத்தாலே this will
Yes,
come
sir.
at least the dot will can itself come under 10 micro 1 micro 1 millimeter. இதில் 1,000 times you divide into this dot into 1,000 times, then you can check about the
Uh,
measurements. Okay, that is all about the measurements. Okay, இன்னும் டெப்த்தாக பார்க்கும்போது the measurement அதிகமாக பார்க்கலாம். So these are
okay, sir.
cytoplasmic extension. Cilia are cytoplasmic extension.
Sir, can you explain that cytoplasmic extension alone again, sir?
Yes, I can explain you. What is this one, Pooja? This one is your plasma membrane, right?
It's a plasma membrane. Yes, sir.
So it is actually the modification of plasma membrane. It is a modification of plasma membrane, but this is their cytoplasm, right?
Yes, sir.
So the contents of cytoplasm is also extended. Okay.
Mm.
So in that case, we can say
Sir.
cilia is a modified plasma membrane, which is an extension of the cytoplasm.
Yes, sir.
Got it?
Yes, sir.
So cilia is considered as a, you can say, it's a modification of plasma membrane and an extension of your
Extension of
cytoplasmic
cytoplasm.
extension. Okay. So we have two types of cilia beta. One is primary cilia and a motile cilia. What I've showed you, everything is a motile cilia. So they
Pencilia.
help in locomotion. Some organisms, the cilia helps in sensory to identify the environment. So we have two types of cilia to understand here. One is your primary cilia, and second one is called as your secondary cilia. Cilia is actually helpful in identification and movement of an organism. For identification, we have a primary cilia, and for movement, we have a motile cilia having same type of functions. Okay, Rama.
Yes, sir.
Now you can see this is an electron micrograph they have given you with the size here. So it's a diagram here, cilia on an ordinary microscope and cilia on a transmission electron microscope. You can see how these modifications are there. So you can
Yes,
clearly
sir.
see here, this one is your plasma membrane. So they are
Mm.
forming the extension of your plasma membrane is your cilia.
Mm.
And the contents of cytoplasm also thrown into it. Right?
Yes, sir.
The contents of cytoplasm also thrown into it, so we're going to call this a cytoplasmic extension. Okay, புரிஞ்சுதாமா?
Yes, sir.
Next is stereocilia. It's an another magnification. Okay, diagram பாருங்க, இங்க பாருங்க,
Mm-hmm.
இங்க பாருங்க. So இது வந்து ஒரு நல்ல ஒரு மேகி நூடுல்ஸ் போட்ட மாதிரி எல்லாம் இருக்கு. We have stereocilia. What do you mean by stereocilia?
I think multiples of cilia, sir.
I'll tell you the stereocilia where they are present means they are found in your inner ear. எங்க இருக்குமா stereocilia? It is there in your
Oh,
inner ear. காதுல இருக்கு.
uh,
What is this cilia பாருங்க? Uh, okay. This is actually a membrane. இங்க வந்து ஒரு மேல வந்து tectoral membrane அப்படின்னு ஒரு membrane இருக்கும். And if here we'll be having some fine thread-like structures, which are called as hair cells. Wait, from the net, I'll just download one diagram to understand this. See, it's an inner part of your ear. Okay, can you see here the inner part of your
I
ear?
yes, sir. Yes, sir.
So in this, you can see here in your inner part of your ear, there is some cells which are called as hair cells.
Sir.
அதாவது முடி மாதிரி இருக்குங்க உள்காதுல இருக்கு நீங்க பார்க்க முடியாதுமா it can be seen only under the normal or
Yes,
electron
sir.
microscope. We have a cell called as hair cells. When sound waves are there na, the sound waves வரும்போது என்ன நடக்கும்னு பாருங்க. When sound waves are coming here, when sound waves are coming here, they'll beat here. The beat பண்ணும்போது என்ன வரும்னாக்கா இங்க beat பண்ணும்போது the இங்க beat பண்ணும். When sound waves are coming in this direction, they go and they will fall on it. This cilia will create some ஒரு தலைமுடி எப்படி நம்ம take your hand and keep it on your head எப்படி ஒரு movement just keep it on your head and check about your I don't know what is your hairstyle, what type of hairstyle you are having, it same like what I saw you in grade 9.
Yes, sir.
So நீ தலைமுடி வச்சு பாருங்க, can you feel the texture of your hair from your
Which-
skin, can you feel your texture?
Yes, sir.
when you just press hard, texture தரல லைட்டாக இருக்கும்போது டெக்ஸ்சர் கொடுக்கும். அதே மாதிரி this hair cells, what they do, they cause one movement called as rippling movement. Because of this
Sir?
only, yes ma,
Nothing, sir.
because of this rippling movement, that is the hair cells, when the sound waves are coming and striking your hair cells, they cause one rippling movement on this membrane, and this signal will go to the brain. So பிரெயினுக்கு வந்துட்டு the hair வந்து ஒரு டோன் மாதிரி போகும். And this is how the keyboards are designed அந்த மாதிரி. This is like a keyboard in your ears. So this will go and they will create some action. That rippling movement will create some signals, and that goes to the inner part of your ear. புரிஞ்சுங்களாமா?
So now the sound waves will fall on the hair cell, sir. The movement, the movement caused by the hair cell is called as rippling effect, which hits that Timberline membrane,
Hmm.
and in turn that from Timberline membrane it goes to the brain,
Tectoral,
sir.
tectoral membrane,
Tectoral membrane.
which creates some electrical signal which is carried into the brain.
Okay, sir.
So it is actually an immobile. They cannot move. Only thing is முடி வந்து இப்படி வந்து அசைக்க முடியாது. Only they can wave movementனால கொஞ்சம் போகும். Like they'll create some wave movement. So this is your
Okay.
hair cells which is found in your ear. காதுல இப்படி இருக்கும். இதுதான் வந்து உங்க காதுல ஒரு மாதிரி sound waves create பண்ணி the waves are carried into the brain. That is the main role of stereocilia. It is a modification of your ear cells.
Okay.
புரிஞ்சுதாமா?
Yes, sir.
புரியலையா புரிஞ்சுங்களா?
I understood, sir.
Okay. Then we have microvilli. What is microvilli? இங்கே கேள்விப்பட்டிருக்கீங்களா? M
Yes,
மீன்
sir.
போடுவாங்க. Yes ma.
Heard it, sir.
Villi somewhere.
Biology only I learned, sir. Can you give any clues, sir?
Okay. Don't think about this for this now. Intestine
Uh,
கேள்விப்பட்டிருக்கீங்களா?
Small intestine, I've heard it, sir.
Okay. So there will have a small confusion always between villi and microvilli. So if you take about villi, if you take small intestine, I have two threads இங்க இருக்கு. One thread இப்படி இருக்கு. One more thread, the thread is like this. Which one is longer? A or B?
B, sir. A.
A. Because the infolding will increase surface area. This is found in the
Yeah.
small intestine, which we are not going to discuss. That is the small infolding of your small intestine is called as villi. But what we are discussing is
Yes,
actually
sir.
microvilli. Microvilli padinaka in a cell, this one is actually your cilia. கொஞ்சம் பெருசா அது thicker cilia. Some finger like projection you can see here like this.
Okay.
Minute projections. This minute projection are called as microvilli. Okay.
Yes, sir.
என்னம்மா?
Yes, sir. So the micro finger projections present within the between cilia.
Yeah, it's a microvilli. It's a cell. It's not
Yes,
an
sir.
organ. So villi is there in your organ, which is small intestine, which we should not discuss. So we're not this. We're not
Okay.
going to discuss about this. We're going to discuss only about this one. In a cell, there's a small projection, which is mostly cylindrical in shape. Okay.
Yes, sir.
So where are they present means they are present mostly on your WBC. If you take your WBC, if you take your dendrites of your neuron, all this area you can see is microvilli, and you can see in your muscle cells இதெல்லாம் நீங்க மைக்ரோவிலை பார்க்க முடியும் சரிங்களா.
Yes, sir.
So what do you think maybe the functions of microvilli? The microvilli are small extension. They increases,
The surface areas.
yes, surface area or to perform efficient function of plasma membrane. பிளாஸ்மா மெம்பரேன் நல்லா எஃபிஷியண்டாக ஒர்க் பண்ணும்போது இந்த மாதிரி பிளாஸ்மா மெம்பரேனோட இந்த மாதிரி பிளாஸ்மா மெம்பரேன் வந்துட்டு will be having more surface area. So the actions of the microvilli will be more in action. புரிஞ்சுங்களா?
Yes, sir.
ஸோ இன்னைக்கு பாருங்க ஸ்டீரோசிலைட் பற்றி பார்த்துருக்கோம். ஓகே, சும்மா ரீகால் மாதிரி we have seen
Okay.
about the lipid raft. Raft is the organization of the lipids with other biomacromolecules. And organization la we are focusing on exchange of information and recognition and cell movement la we are focusing on cellular movement. For this we are going to discuss about cilia, a small extension, or you can say a modification of a plasma membrane having some cytoplasmic extension. And the cilia is of two different types: primary cilia, which helps in sensing the environment; secondary cilia or the motile cilia helps in movement. And stereocilia will not help in movement, but they help in sensory. Am I correct?
Oh, yes, sir.
Microvilli also, they will are involved in not movement, they are involved in sensory. புரிஞ்சாமா?
Yes, sir.
Now the diagram பாருங்க. சும்மா just have a basic outline of this diagram. சும்மா ஒரு நிமிஷம் பாருங்க. பாத்தீங்களா?
Yes, sir.
Okay. Now what I have to draw is I'm going to draw one diagram to understand this. Yeah. Am I taking longer time to complete your topic?
No, sir. This is enough, sir.
So this is your microvilli. Okay. This one is your microvilli. இதுல ஒரு சின்ன ஒரு modification பண்ணும். After April, I'll be free, Vanidama, because students are exams are getting over by April. College students,
Okay,
yeah,
sir.
I have some college final year students. So for them, the course is
Okay.
getting over by April. So April
Okay,
23rd
sir.
is their zoology examination. Okay.
I have no problem, sir, because the school only starts in the month of September.
அதுக்கு முன்னாடி முடிச்சிடும் பூஜா. ரொம்ப டைம் எடுக்க மாட்டேன், சரிங்களா?
Okay, sir. Okay.
Okay. Let me label the parts here. Ma, this is cell A would a component, this cell
Mm.
and another cell component. சரியாமா? Okay, so this one is your microvilli, a small modification of your plasma membrane. And
Sir.
this is, okay, I can say as adjacent cell membrane, that is between the two membrane, the connecting link, the adjacent cell membrane. Okay, now, okay, I'm going to take one modification here. In the cell-க்கும் cell-க்கும் இங்க பாத்தீங்கன்னா there are some modifications which are called as cell junctions.
See. Okay. S- ah,
So cell junction is a junction which connects two different cells are called as your cell junction. Okay,
yes, sir.
what a cell junction means? It's a junction which connects two
Con-
different cells
connects two different cells.
are called as your cell junctions. அப்படின்னு சொல்லுவாங்க.
Cell
Now
junctions.
there are a lot of cell junctions are there. Okay, so if you look about here, here we have some junctions, and I'll just magnify here the same junctions. I'll show in a different way. இங்க tight junction, adhering, desmosin போட்டிருக்காங்க. Let me go with that junctions now. Okay ma, இதே
Yes,
diagram
sir.
with small modification. Let me draw it here.
Mm.
So on the complete cell எடுக்கிறோம், இங்க complete cell எடுக்கிறோம். So this is the inter our adjacent cell membranes are there here. It's a cell membrane. It's another cell membrane.
Mm.
Now there's one junction. This area alone. So two cells have to be held together, right? In one particular area,
Yes, sir. So the separation part between the cell and cell B, that rectangle is called as
it's adjacent cell
adjacent
membrane.
cell membrane, sir.
It's the adjacent
Okay.
cell membrane. Okay. This one is called as your tight junction. What is this one called as?
Tight junction.
This one commonly we're going to call a junction called as tight junction. So here what happened? Adjacent cells are fused without any spaces, so they are keeping the cells very close to each other. So how this entire structure of cell gets modified? Let me take my eraser. இப்போ வந்து eraser வச்சு மறைக்கும் போது this cell are going to get pulled together. இப்படி ரெண்டு பக்கம் பக்கம் வந்துரும். They'll pull the two cells components together. இப்படி கிட்ட கிட்ட இழுத்துரும். புரியுதுங்களா?
Yes, sir.
This is called tight junction. In tight junction there will not be any spaces. இது அப்படியே நம்ம proceed பண்ணிட்டு அப்படியே விட்டோம்னாக்கா, I'll just it as this. இது அப்படியே நம்ம proceed பண்ணாத அப்படியே விட்டுட்டோம்னாக்கா, they will be like this. This will be completely fused. ரெண்டும் fuse ஆகி இப்படி வந்துரும். So there is no space between them. This junction is called as tight junction. Purinchama?
Yes,
That is
sir.
only shown here as zona occludens. அப்படின்னு சொல்லி போட்டிருக்கிறாங்க. Zona occludens. This one.
Okay.
அப்போ how they are bound எப்படின்னா இங்க ஒரு protein இருக்கும். அது பேர் வந்து occludin protein. The name of the protein, this is a junction, the space per junction. அப்ப இங்க லைனா வரைஞ்சிருக்கேன்ல, what is this one? This one is called as occludin protein.
The dot with red line connecting the both
Yeah,
cells,
not with
sir.
dot with all this extension cylindrical structure,
Uh,
that is called occludin protein. So what is the role of this occluding protein? They act like a clip. You have a normal clip. To join the papers and all, we can use stapler machine. So tight up பிடிச்சிக்க மாதிரி ரெண்டு செல் பக்கத்து பக்கத்துல கொண்டு வந்து they make it that's a very tight. புரிஞ்சுதுங்களாமா?
yes, sir.
This one is called as occludin protein which
Mm-hmm.
helps in tight junction. இது புரிஞ்சாமா?
Yes,
So
sir.
tight junction just keeps the cell to cell between contact. No exchange of, because the topic I'll be discussing is cell movement பார்த்துட்டோம், exchange of information பார்க்கணும், கரெக்ட்டுங்களா
Mm.
இல்லையா? We are focusing on that.
Yes, sir.
So they will not help in any movement. The movement of molecules will not take place in this. Just the occluding protein just adheres and keeps the two cells together. Purinchinglamma?
Together.
So
Yes,
I'm going
sir.
to take another set of protein. Okay, not red color. I'll take blue color here. கொஞ்சம் bold line எடுத்துக்கலாம்.
Sir, now you're explaining the junction systems now, sir.
Yes, ma'am.
Always within the within the plasma membrane, will they always be present? Will they always be in
all
the form of tight
all
junctions?
இங்க four port இருக்கு. All will not be there together.
Ah,
Only one can be there or two can be there,
ah,
but generally these are generally we can see the following junctions.
okay.
So
Any
now,
one or two will always be present in the plasma membrane.
yes ma, sometimes
Okay,
all four may be there, depends upon what type of cell it is.
okay.
So this junction is called as adhering junction. What is this junction called as, beta? Adhering
Adhering junction.
junctions. Adhering அப்படின்னா just joining. It's just keep the two cells together. வேற எதுவுமே பண்ணாது. Interlocking மட்டும் நடக்கும். And there will be an exchange of molecules because between this there are some free spaces, right? இங்க ஃபுல்லா block ஆகி கிடக்குது. But there are
Yes.
some free spaces,
Ah,
right? This is bound by a
yes,
protein
sir.
called as cadherin protein. The protein which binds this adhering junction is called as cadherin protein. Purinchama.
Yes, sir.
Next we have another one called as desmosis. இது எதுவுமே செல் எதுவுமே பண்ணல பார் செல்லுடைய அந்த டிஃபரன்ஸ் பத்திரமா வச்சிருப்பாங்க. There will not be any change in that. So I'm drawing and showing you to make you to understand. Shariyama?
Yes, sir.
So have you got enough friends over there in France?
And in my neighborhood, I have, sir.
Hmm.
But in the university, I just went for 1 day, sir.
அடடா அதுக்கு ரெஸ்ட்
Yes, sir.
அதுக்கப்புறம்
I went. Monday I went to school, sir.
Hmm.
The issue what happened there is the entire school was smoking.
Hmm.
So so I don't have asthma, but I started having some breathing problems, sir. So with that breathing problems, as I was not able to breathe properly, I started fainting.
Hmm. So this you're going to have a change in your college.
The no. The professor over there said that let her come and join the upcoming year, the next year, sir.
Hmm.
Uh,
Okay. Now listen here. It's actually like a welding protein. It's a transmembrane linker. It's called as desmosis. We used to use a term called desmosis. This also helps in joining two different cells. அவ்வளோதான். இது வந்து என்ன பண்ணாக்கா ரெண்டு செல்ஸை வந்து பக்கம் பக்கம் இருக்க யூஸ் பண்ண அவ்வளோதான். ஸோ here we have. So they help in they have the same function like adhering, but it's another tight connection. இது கொஞ்சம் டைட்டா இருக்கும் உங்களுக்கு கொஞ்சம். So what is the main thing in this? The intercellular spaces are more in number and helps in transport of molecules here also. And here some fibrils are there. These fibrils are called as tonofibrils. For your understanding, it's not in your content here. This fibril is called as tonofibril. அப்படின்னுவாங்க. The help of tonofibril, they are joined. These are additional supportive structures along with the adhering junctions. Okay, ma?
Sir,
Here
can you
also the
repeat this alone again, sir?
Enama? Enama? Come on, tell me da.
Sorry, sir. Can you repeat desmosis alone again, sir?
Okay, I'll tell you what is desmosis. In other way, I can
Mm.
say it is there in your heart beta. On the skin, just you just skin கொஞ்சம் இழுத்தாலும் it will come back to the normal position, right?
Yes, sir.
So it is mostly found in your plasma membrane of your skin. Desmosis enaka. There are some. I didn't use any fibers here. I use a fibril here, right? So any mechanical
Yes.
stress, like we are just peel off your skin, just skin கொஞ்சம் கிள்ளி பாருங்க, நான் இழுத்து பாருங்க. They come back to the normal
Mm.
position. ஒரு குறிப்பிட்ட ஸ்டேட்க்கு அப்புறம் you cannot pull your
Yes,
skin
sir.
back, right? It's
Yes,
because
sir.
of between your skin cells we have desmosis with tonofibril, which gives them mechanical support and which can help the cell to resist mechanical stress. And they give you the mechanical support. That is the function of desmosis. Can you see the desmosis here? And the fibers மாற இங்க கொடுத்துருக்காங்க
Yes,
பாத்தீங்களா?
sir. Ah.
இதையும் கொடுத்துருக்காங்க. This one is called as desmosis. புரிஞ்சுதுங்களா?
Sir, the joining of cell
Hmm.
takes place with the help of the desmoglein protein and the fiber-like structure is called as tunofibril.
Tono.
Tono. Okay.
Okay, I'll write very clear, not earlier. Tono. 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 It's a tono fiber. Okay, Ramma?
Sir.
Okay. And the help of tonofibril, they give you the mechanical support and saves the cells from mechanical stress. So cells இழுக்க பிடிக்கும் போது செல் நாச ஆகாத இருக்க other support can be broken, but this is way tough to be broken. புரிஞ்சுதாம்மா?
Yes, sir.
Last but not the least, we have another important junction. This one is called as gap junction, which is not shown in the given diagram. This junction is called as gap junction. It says that this is a space where it's completely gap. So through this, exchange of molecules can take place very easily with the help of this protein pipes. So these two junctions are there. It acts like a channels through which you can see that exchange of molecules can take place. Yes or no?
Yes, sir.
That is called gap junction. So there are four important junctions. One is called as tight junction, adhering junction, desmosis, and gap junction. In Greek, it's called zona means zones, occludence, tight, adherence, that is to join. Desmosis is actually a Latin word, and gap is an English word. Okay, ma?
Yes, sir.
It's very important. The junction system are very important. Then in 11th CBSE, you told you are NAOs, right?
No, sir. 11th I was CBSE, sir.
அங்க செல்
But the
எடுக்கலையாடாமா?
teacher did not teach. No, sir. She just taught us like two chapters, sir.
No problem, da. I'm here. Now you are able to understand about the four type of junctions, right? Because for
Yes,
students, I will
yes,
not say
yes,
about
sir.
the protein names since you are there in the college. So I'm saying you about the protein names. Okay.
Sir, in gap junction, what is that protein name, sir?
We don't have a specific protein for them, Dhamma.
Okay, okay.
Okay. But some people say
Okay.
we shouldn't get confused with this protein here. This is a tonofibril comes for this.
Yes, sir.
People say it is actually a connection pipe அப்படின்னு வாங்குது பேரு.
Mm,
And the name of the protein is connexin அப்படின்னு வாங்க.
mm.
Connexin 43 அப்படின்னு ஒரு protein இருக்கு. I think it's 43. Connexin protein just connects. It's called connection pipe. Through this, exchange of
Okay.
molecules can take place. So there are four important junction: tight junction, adhering junction,
Junction.
desmosis, and
Responses.
joint junction.
Gap
இதுல
junction.
tight junction adhering keeps the cells strong with each other. Desmosis gives them mechanical support. Gap junction they help in transport of molecules. புரிஞ்சுதாமா?
Yes, sir.
The weakest among them is the gap junction.
Gap junction. So in adhering junction, does now you said that it keeps the molecule together. So does the exchange of molecules take place, sir?
It will not take place in the tight junction. In others, the exchange of molecules will take place, but it's highly
Take place.
limited.
Okay.
புரிஞ்சுதாமா?
Yes, sir.
ஸோ உனக்கு வந்து ஆகுமா ஆகாது அப்படின்னு சொன்னாக்கா எஸ் ஐ கேன் டெல் யூ வெரி ஈஸிலி that in case of the first junction that is called tight junction, tight junctions, I think I can give you the points here. I'll type the points here for tight. Are you able to understand what is tight junction, right?
Yes, sir. Yes, sir.
When you go with tight junction, so in tight junction what happens is it keeps the two cell membrane together. No transport of molecules takes place except selectively permeable substance molecules. You know what is selectively permeable? Selected molecules can cross through, not other molecules. புரிஞ்சுதாமா?
Okay, sir. Yes, sir.
Next one is adhering junctions. Um, just keeps the two cells intact, that is பக்க பக்கம் அப்படியே வச்சுக்கும். And here transport of molecules takes place. Next is desmosis. Welding or cell. Two adjacent cell membrane together and gives mechanical support as well as transport of molecules. Here also the molecules will get transported. Okay, ma?
Yes, sir.
Gap junctions, tunnel-like protein pipes and allows most of the molecules to be exchanged fast. Purinchama?
Yes, sir, in adding you place, you have add an extra n, sir.
Okay.
Uh,
Got it. This is my own thing for your understanding. I've given about the four types of junctions.
yes,
Okay.
sir.
To understand that, இங்க
Yes,
பாருங்க
sir.
டைட்டா இருக்கும் போது molecules pass ஆகாது. பாக்கி எல்லாத்தையும்
Pass
molecules கொஞ்சம்
ஆகாது.
pass ஆகும். In all the molecule passes except the tight junction. This just இங்க பாருங்க just the keeping except the tight junction மட்டும் தான் ரெண்டு செல் கிட்ட கிட்ட வந்திருக்கு கரெக்டா?
Yes, sir.
Only in tight junction the cells are together. In other junction cells are not together. Just they keep them intact. Other junction only they keeping the cells so close to each other. But here it's not like that.
Yes, sir.
Okay.
Understood, sir.
Yeah. So we have completed up to this. So next class we'll focus on hemi. The type of desmosis called hemidesmosis is also there with us. Then how I told you
Okay.
the molecules are getting transported. So next class
Yes,
it will
sir.
be how they are getting transported. That will be the next class, which will take again two more lectures. Maybe by Saturday we'll be able to complete the types of transport. Okay.
Yes,
So
sir.
we'll be able to finish up to membrane transporter mudikkum. Active will take some time. So I think hope you can
Yes, sir.
understand that up to this junction system I have completed. So the two important
Yes, sir.
topics I have completed for you. Okay.
Yes, sir.
Later when the class gets completed, it will stop recording and I'll stop the recording too.